Okay, in this video, I'm going to look at the process of protein synthesis. So we're going to look at the protein synthesis in terms of transcription and translation. The idea being the cell wants to produce a polypeptide from just one gene of a selection of genes. What it's going to have to do is it's going to have to copy that section of DNA, then send out a copy to the cytoplasm, to the ribosomes, where that copy can be decoded to produce your actual polypeptide chain, your amino acid sequence. So if we look at the process of transcription first, so transcription takes place inside the nucleus. So you start off with your piece of DNA. And the first thing that's going to have to happen to your piece of DNA is you're going to have to actually separate that piece so that the two strands will separate. The enzyme that does this is DNA helicase. And DNA helicase breaks hydrogen bonds. This is one of the few times that you're allowed to use the word breaks in terms of a bond, because it's not a covalent bond, it's a hydrogen bond, which is a bond holding the complementary bases together. So our DNA will unwind. And it will expose particular complementary bases. So, for example, A, C, G, T, G, C. The next stage that will happen is that complementary RNA nucleotides will line up due to the base pairing. So if I have a look at my piece of DNA, and I have my sequence, T, G, C, A, and I've got my sequence up here as well. The complementary RNA nucleotides will come and line up, but they will only line up along one strand, which can be called the template or sense strand. So the nucleotides will line themselves up. You've got A, you've got C, you've got G, and of course you have U instead of T. Now you will notice that the nucleotides are not joined together. We've not made the sugar phosphate backbone. And that's because to do that, you need the enzyme RNA polymerase. So the next thing that will happen is that RNA polymerase will catalyze the formation of the phosphodiester bonds between adjacent nucleotides. And it's really important that you do know the job of RNA polymerase. It's not always clearly given in books. So if I go back to my picture, I have my piece of DNA still, like so. I have my bases. I'm just going to worry about the ones that are down here for now. I have my nucleotides, which were there. And just to show you in a different color, the phosphodiester bond, that's what's being catalyzed by RNA polymerase. And that will form what we call pre-mRNA. because it's not quite the finished article. All that will happen once it gets to the end of the gene is that the DNA will wind itself back up again, and you'll be left with your pre-mRNA. You are sometimes asked to compare transcription with DNA replication. So there are certain things and certain similarities. So for example, both transcription and DNA replication use DNA helicase. Both of them have complementary nucleotides line up by base pairing. However, DNA replication has DNA nucleotides and transcription has RNA nucleotides. In DNA replication, both strands are copied, whereas in transcription, you only copy the template or sent strand. And lastly, they both use polymerase, but transcription uses RNA polymerase, and DNA replication uses DNA polymerase. So what is pre-mRNA? Well, if you actually look at messenger RNA, what we know is we can divide it into what we call introns and exons. And we'll label the exons 1, 2, and 3. Because in order to make pre-mRNA functioning as mRNA, what you have to do is you have to splice out those introns, which means you effectively have to remove them. And then you join the exons together, like that. So number 1, number 2, number 3. Now, the beauty with this is you could join them in a different order, 1, 3, 2, 2, 1, 3, which means that one pre-mRNA can make several different mRNAs, which can therefore code for several different sequences. Now, next thing we have to think about is translation. Now, before we think about translation, we need to understand the genetic code. There are 4 different bases that make up DNA, and we know that they are arranged in triplets, which means that there are 64 possible combinations, because 4 times 4 times 4 is 64. Feel free to sit down and write A, C, G, A, G, C, etc., or just have a look at the table. You'll see there's 64 different combinations. But there are only approximately 20 amino acids that need to be coded for by my 64 triplets. Now, what that essentially means is that there are several different triplets that code for the same amino acid. This term means degenerate. That's what degenerate means. It means several triplets code for the same amino acid. So, for example, it may be that A, C, G, U may all code for the same amino acid. What else do we know about the code? We know it's universal, which means that is the same in all organisms, prokaryotes or eukaryotes. And we know that it is non-overlapping. That means that a base can only belong to one triplet. So for example, if you have the following bases laid out, you can literally split it into triplets because each base can only belong to one triplet. So there is a table which connects each triplet to its amino acid. You do not need to know that table. You'll be given it in a question. The only thing you need to be careful of is what the table is showing you. Sometimes it will show you a DNA triplet. Sometimes it will show you a messenger RNA triplet. And sometimes, as we shall see, it will show you a tRNA triplet. So what is happening in the actual process of translation? So the first thing that happens is that the messenger RNA joins a ribosome. And when it joins a ribosome, two triplets, or codons as we call them on messenger RNA, are exposed. So what do I mean by that? Well, let's imagine a picture. Let's have a picture. We've got our messenger RNA over here. A, G, U, U, A, C, U, U, A. I'll put one at the beginning. A, U, G, because that's always the start codon. C, C, C. So the ribosome will come along and it will join with its large subunit over the top, like so, and its small subunit along the bottom, a little bit like a bun wrapping the burger or the lettuce, whatever you want, okay, exposing two codons at a time. Okay, now sitting in the cytoplasm, sitting in the cytoplasm, there will be transfer RNAs. Now, transfer RNAs are RNAs that form into this sort of clover shape. They're still single-stranded, but they form hydrogen bonds between them. And at the bottom of this clover shape are three bases, which we call an anticodon. And depending on what those three bases are, it will carry a specific amino acid. We'll say amino acid number 1. Okay, so there are 64 different transfer RNAs, all carrying their specific amino acid. So what's going to happen? Well, the correct transfer RNA is going to come in. It's got to be the correct one that has the corresponding anticodon. So in this particular case, it will be U, A, C. And the second one will also bind, which will be U, C, A. And they will both be carrying their amino acid. We'll call that amino acid number 1, just to make life easy. Wrap that out, give us a bit more space. Okay, and we'll call that one amino acid number 2. So we have our 2 transfer RNAs. That's the next stage. So we had our messenger RNA joining with our ribosome. We had the exposure of 2 codons. And then when you're writing what happens in the next stage, I'll just write it underneath for you. You've got it written down. You say that 2 tRNAs, each with their specific, and this word specific is really important, so I'm going to underline it. It's underlined in mark schemes. Pair with the messenger RNA so that their codons, or so their anticodons, form a complementary base pair with the mRNA codon. Okay, so we've got 2 tRNAs. They've paired up with their anticodons pairing with the codon. And the next thing that will happen is that our amino acids that are up here, they will bond they will form a peptide bond. Okay, so that's going to be number 4, is that a peptide bond is formed between amino acid number 1 and amino acid number 2. Redraw in my ribosome. Okay, so peptide bond forms between the amino acids. And the first tRNA will leave. It will leave to pick up another amino acid. So my first transfer RNA, which is this pink one over here, that will now go off. Okay. And that will now go off to pick up another amino acid. And the whole ribosome will now move on. Okay, so if we move the whole ribosome will now move on by one codon like this. There we go. And the same thing will happen. So my next transfer RNA will come in, like so. With its amino acid, my peptide bond will form, like then, and my next transfer RNA, that transfer RNA will now go off. And the whole ribosome will move on again, like this. And it will keep going all the way until it reaches what we call the stop codon, which is where the ribosome knows that the sequence has come to an end. So if we write that underneath, love that out. Okay, we can say that the ribosome moves along one codon and repeats until the stop codon is reached. And then finally, polypeptide is formed. And that's pretty much the end of translation. So there's just a couple of things to be aware of that you need to be sort of keeping a watch out. So for example, a couple of things that they sometimes ask you. So they'll give you a DNA sequence, then they'll give you a messenger RNA sequence, and then they'll give you a tRNA sequence, and maybe they'll give you the amino acid sequence. So for example, that they'll ask you then to fill in the messenger RNA. Remember, it's going to be complementary, but you mustn't have a T in there. So that would be U. Oops, mistake there. A, U, U. And then fill in your tRNA sequence. Now, tRNA sequence will be the same as the DNA sequence, but again, I'm not allowed any T's, so I've got to switch it to a U. And then if they ask you an amino acid sequence, they will give you a table. You just need to be careful and look at the table to see whether the table talks about mRNA codons or tRNA anticodons, so that you can determine what you're looking at. The other thing they often ask you, so sort of interesting question is, let's say I have my DNA, my gene is 150 bases long. First question could be, what is the maximum number of amino acids? Now that would be 150 divided by 3, which is 50. So it could be 50 amino acids long. And then it will say, but why is it less? Now, it could be less for a number of reasons. It could be because that 150 bases also contains some introns. It will also likely to be because that 150 bases will include a start codon and a stop codon, and the start and the stop codon are not used in the final polypeptide chain. So those are things to be aware of when we're talking about protein synthesis. You need to understand the genetic code in order to understand mutations, because the different types of mutations will affect the amino acid sequence depending on where the mutation is. So for example, a mutation in the third base of a triplet often has no effect, although it could sometimes cause the wrong amino acid to be coded for. A substitution in base 1 or 2 of the triplet will always cause the wrong amino acid to be coded for, or nearly always, unless you're very lucky. And an insertion or deletion, that will cause all these triplets to change from that base onwards and cause what we call a frame shift mutation, where the whole sequence changes. We'll talk about mutations in another video as well.